Morning everybody. I welcome you all back, and as we did brophytes last time, and now today I will start the algae. So algae is basically its expandable form, so I will take class by class, and today I will take the chlorophyce and subsequently the other classes on the subsequent days. So you have to refresh your knowledge today of chlorophyce, and when I will be back tomorrow, so you can ask me the questions and you should have refreshed chlorophyce. Then I move to the next class, because algae is a very important and you have to read every day, otherwise it is very difficult to understand algae. So right now, be indoors, be safe. So remain with me. I'm coming back in few minutes. Bye bye. Morning everybody, welcome back. And today I am taking you the word of fascinating algae. So algae, which is basically comprised of group of chlorophyll containing thylakoid plants which are placed in division Thallophyta, and the algae word comes from the Latin alga, which means seaweed. Algae are further classified into the aquatic algae, and aquatic algae consists of free floating, in which Chlamydomonas, Volvox and Spirogyra. And today I am taking the Chlamydomonas. Attached we have Oedogonium, Ulothrix and Holdfast. And today we will taking the Oedogonium. And these have all their cells side filament having chlorophyll, except the last one, which helps in attaching to the substratum. It is called Holdfast, right? And then the third types is the cryophytic algae. Cryophytic algae is the algae which is growing ice and snow. The best example is Chlamydomonas nivalis. Chlamydomonas nivalis gives red color to the snow due to pigment called hematochrome, right? And the fourth type is the parasitic algae. Parasitic algae, it consists of Cephalurus virescens. And this Cephalurus virescens cause the red rust of tea, right? And it is a parasitic algae. It causes havoc to the tea plants in Assam tea garden. This Cephalurus virens, right? Besides this, parasitic algae also attacks coffee, mango, rhododendron, right? So because of the space constraint, so it is not possible me to take all the types, but there are other types, like first is benthophytes. Benthophytes, they are algae found in attached conditions. And here examples are Chara. We are taking Chara this time, today, right? Then we have Cladophora and Gangrosira. Later on I will talk about this Gangrosira. So remember, I will talk with some algae as a Gangrosira stage, because it resembles to the Gangrosira algae. So then next is the fluviatile. This fluviatile algae, they are found rapidly in flowing water, right? So these vital algae are basically Ulothrix, which are present in the mountain falls, and then is the Batrachospermum, which are in the swift running streams of Dehradun. I just want to take it again when I do the rhodophyce, but I tell you that in case of rhodophyce, all are the marine algae except Batrachospermum, which we read in the BSc. It is a freshwater, and here I will say it is in the running streams. And the next type is epiphyllophytes. Epiphyllophytes is the algae which are present in the bark of trees. And here the example is Trentifolia. Trentifolia is a very important, because it will come in paper in the form of which of the following is the aerial alga. So Trentifolia is the aerial alga also, right? So now I will come directly to the our next first class, that is chlorophyce. Chlorophyce are predominantly freshwater, about 90%. And the main pigments are chlorophyll A, right? And chlorophyll B. And they are found on the thylakoids lamellae of chloroplast, right? The shape of chloroplast are cup-shaped, which we are doing today, Chlamydomonas, girdle-shaped, Ulothrix. One question will come from here. Reticulate, Cladophora, Oedogonium we are doing today. Spiral, same from the word, Spirogyra, and discoid, Vaucheria and Cara. And Cara we are doing today, right? So reserve food is starch in case of chlorophyce is composed of amylose and amylopectin. And they have well organized nucleus, but when we do synaphysis, I will say they are not well organized. They don't have the membrane, outer membrane, that is nuclear envelope. So that I will take later on stages. So the first genera I will take is the Chlamydomonas. So Chlamydomonas is basically unicellular, freshwater, motile alga. So this one question will come, and it provides green turbidity to the ponds. The green color or to the ponds, they are because of the Chlamydomonas, right? So here I want to mention you one thing, that is basically there are different genera besides Chlamydomonas, like Coleochaete. Coleochaete grows on the aquatic plants and grasses, right? Then we have Cladophora. Cladophora, it grows on the mollusk shell, mollusk. Then Tentifolia, I told you already, it is aerial alga. And Chlamydomonas nivalis, I already told you, it is on the snow, and it gives basically red color to the snow. That is due to the pigment called hematochrome, right? So now I'm coming to again to the Chlamydomonas. So Chlamydomonas, one variety is Yellowstonensis. Chlamydomonas yellowstonensis grows on the snow-covered mountains of Yellowstone National Park of America, from where they will get the name, right? Then is nivalis, I already told you, imparts red color to the snow due to presence of red color pigment hematochrome, and the chloroplast has a single paranoid. Now question is very important, how the Chlamydomonas looks like. See, this is Chlamydomonas, and this Chlamydomonas, they have this cup-shaped structure, right? So when we do Chlamydomonas, and here we have flagella, and these flagella arise from the two dots called blepharoplast, and these blepharoplasts, they are attached by means of paradesmos, and these paradesmos from basically attach the blepharoplast, and these blepharoplasts, they are basically the origin of the granules, starch granules for the flagella. And they give rise to flagella, and flagella, the lowermost part is thick, it is called axoneme, whereas the upper last part is naked, it is called endpiece. And this blepharoplast, one blepharoplast attached to the centrosome of nucleus, and this by means of this long fiber called rhizoplast, right? And this whole structure is called neuromotor or flagella apparatus. So these questions will come as a MCQ also. Then below them there is a paranoid body embedded in it. So, and this paranoid body is already covered outside by means of a starch sheath. Whenever the starch sheath is incomplete around the paranoid body, the structure is called polar paranoid. So this is very important to know what is polar paranoid and why it is called polar paranoid, because starch sheath is not complete. And here it is very important to tell you that in case of chloroplast in Chlamydomonas is basin shaped, BASIN, basin shaped, and chloroplast of chlorophyce have paranoids, except Cara and Vaucheria, which of this the Cara I will take today and Vaucheria in the subsequent days, right? And I already told these paranoids are surrounded by starch sheath, and these paranoids are crystallite in structure, and that is why these paranoids, they are called pyrenocrystals, P-Y-R-E-N-O, pyrenocrystals, right? This I already told you, when the starch sheath is incomplete, it is called polar paranoid, right? So here today I refresh you again. We did rhizoplast, a long fiber connected to the centrosome inside nucleus, and just below the nucleus is present nucleolus, right? And embedded below is the starch sheath, right? And the starch sheath which is covering the paranoid, right? So this is the structure we will getting, and the starch sheath incomplete around the paranoid, and that structure is called polar paranoid, right? So now we are coming to the reproduction part. and the reproduction part is basically the aplanospores, right, by means of aplanospores. So in case of reproduction part, so when we will do reproduction part, so by aplanospores there, here it is very important to remember that these aplanospores are non-motile cells, right? Very important. This formed during the unfavorable condition. As I already told you, aplanospores create a thick wall around them and they become hypnospores. So in case of Chlamydomonas nivalis, and these hypnospores, they all cells of together forms a palmella stage. In C. nivalis, all the cells of palmella stage create a thick wall called hypnospores, and these hypnospores are red in color. I already told you, due to the hematochrome or red pigment. Hence, the red color of snow is due to the red color in the hypnospores, which we did right now, just a moment, right? So now we are coming to the next part, that is the sexual reproduction part, right? So in sexual reproduction part, I have to take you back to the introduction part. First, it is the isogamy. When two gametes which are morphologically and physiologically similar are called isogametes, and it is in case of Eulothrix and Chlamydomonas, which we are doing right now. And the second type is anisogamy. In anisogamy, the one gamete, like female, is bigger than that of male, and that is the characteristic which is anisogamy. And third type is oogamy. Chlamydomonas, it is a most advanced type and large non-motile egg fused with the small motile sperm, except in case of rhodophyceae where the sperms are non-motile. Please remember this, and this is very important, right? So this we finishes the Chlamydomonas, and I am moving to the next part, that is the Oedogonium. Oedogonium is basically a freshwater alga, and see, thallus is multicellular and filamentous, and the filaments are unbranched, right? Take care of these things like multicellular, unbranched, uniseriate. The basal cells which act as holdfast is devoid of chloroplast. I already told you right now, right? So filaments, they are long and unbranched, and the basal cell of the young filament is devoid of chlorophyll, and attached to some substratum is called holdfast. And later on I will tell you when I will do that it is a holdfast which does not do the reproduction part. Except holdfast, all cell will do the reproduction, right? So now we are coming to the reproduction. Before reproduction, I will tell you the chloroplast. I already told you earlier in introduction, reticulate. Pyrannites are present with starch sheet around, as in case of Chlamydomonas, right? So, like this. Same, right? Then when the sheet is incomplete, is called polar pyrannoid. So now we are coming to the next part, that is the asexual reproduction. Before that, I move to the vegetative reproduction, that is fragmentation, small fragments, then akineids. Akineids here are under unfavorable conditions. These thick wall structures are formed, and these thick wall structures are brownish in structures, and they are called brownish structures, and they are called akineids. And then the asexual reproduction. Asexual reproduction is very important. Here the all cells, mind it, all cells except holdfast are capable of producing zoospores, right? And zoospores which are produced within these cells usually rich in food reserves, and this cell is called zoosporangium, right? So zoospores, they are produced in zoosporangium, and these zoospores, they results in the asexual reproduction. So sexual reproduction, we are coming to the oogamous type. I already told you it is the most advanced type. Here the large non-motile egg fused with the small motile sperm, except in case of rhodophyceae where the sperms are non-motile. But here the oedogonium, it is a very characteristic feature. Here it is two type. I will take one by one. First is Macrandrus species. In these species, Antheridia are born on filaments of normal size, and these Macrandrus species, they are basically two types: homothelic or monoecious, where antheridia and oogonium are formed on same filament, whereas second type is heterothelic or dioecious, where the both are produced on the different filament. And now I will do the second part. It is the Nenandra species, right? In Nenandra species, basically here I just want to add, because the Nenandra species male filaments are smaller than female, and they are called dwarf males or Nenandrium, right? So Nenandra species are always dioecious. Please mind it. So if a question comes, then Nenandra species, so it is always dioecious, right? In Nenandra species, antheridia develop on specialized small 2-4 celled filament known as dwarf male. The dwarf male develops from the androspores, which are formed in androsporangia, right? So basically dwarf males, again revise you, or Nenandria, they originate from the germination of special type of swarmers, SWARMER swarmers, known as androspores. The androspores, they are produced in androsporangia. And these, how the androsporangias are formed? Androsporangia are formed from the ordinary vegetative cell due to, please mind it, transverse division. This question will come. Which division? It will give option to transfer, longitudinal, both or none. You will add it is a transfer divisions, right? Where the androsporangium and oogonium develop on same filament is called gynandrosporous. This is very important, right? When the two are born on the different, it is called idioandrosporous, right? So gynandrosporous or idioandrosporous, the two types you should be knowing. So now we are coming to the structures. Three structures we did is the zoospores in asexual. Then we have the antherozoids and androsomes. See this. Structure and shape of zoospores, androspores and antherozoids are same. But the difference in them is the size. Size means zoospores are larger, androspores are smaller than zoospores, medium size, and antherozoids, they are small in size. Antherozoids, they are also differ from the androspores just because of the lesser number of the flagella. And here I will move on to bookish part, that is the see, this is Chlamydia structure, and this is, I did the anisogamy type, right? And now we are moving to the next part, that is the oogonium, right? This is the oogonium. Oogonium, you will see caps. Then we have chloroplast. Then we have pyronite, and these are capsules. This question will come. And then we have the chain of antheridias. And last is the holdfast, right? So now this is the oogonium we did. Just I told you that I showed the diagram. So and this oogonium basically is responsible for the formation of cap cells, right? So now I am coming to the next, that is the formation of zoospores. See, these are the how the zoospores, I already told you, they are formed. And now we are coming to the formation, this development of antheridia, and the antherozoids, they are formed, the 2 in number. They are present in the vesicle. Now this I already done is the Nenandrous species, and this is the dwarf male, right? This is dwarf male, right? And here I will tell you, these are supporting cells, but they don't come by the name of supporting cells. They are come by the name of suffultory cells, SUFFULTORY. So they come by means of that. So now we finishes this, and this is the cycle, and as we know, cycle is dominant by the haploids, that is, it is haplontic. So now we are coming to the Kara next, also called stonewort. Why it is called stonewort? Question, a lot of time question comes due to the calcium and magnesium carbonate. It is same that of like oedogonium, it is multicellular. But here, one thing will come, you will have to remember, macroscopic. So macroscopic is the this genera basically which is bigger in size, and this, right? It differentiates into rhizoids and main axis, right? Rhizoids and main axis. Rhizoids are white thread-like multicellular. First is white, then multicellular, uniseriate, and branch structure with oblique septa. White, and two things we will remember: white and oblique septa. Main axis is a long branch differentiated into nodes and internodes, and the cells originating at nodes are called cortical cells, right? So just be attentive. Half of the cortical cells grows upwards, means lower half will grow upward, question will come, and other half, that is downward, upper half will go downward. So to explain this, I will just tell you, this is the structure of Kara, and this structure of Kara is made up of this main axis and there is a rhizoids. Main axis means nodes, internodes, nodes, internodes, and this main axis is because of nodes and internodes, and it have rhizoids, which I told you already. It is of white color. And now we are coming to the next part, that is C, which I am telling you. Here there are two filaments. Upper one, they are moving downward, they are called descending. The lower one, they are moving upward, they are called ascending, right? And from the lower node, you will see the secondary laterals are coming, right? And these also arises from here. They are secondary laterals called stipuloids, right? So we are back again to our point, which I told you. Diagram I also show. Half of the cortical cells grow upward, lower half, and other it will downward, upper half. So stipuloids, the basal nodes of branches of limited growth, develop some unicellular outgrowth, which just show you, right? They are called stipuloids. Some species of Kara, that is Kara nuda, are unistipulate. This is very important. Question will come from here. Unistipulate, number of stipulate at each node is equal to the number of primary laterals, because I already told you from lower node there we are coming the primary lateral and stipuloids. But most of the species, they are bistipulate. The number of stipulates at each node is twice. Bi means do, two, right? So now we are coming to the vegetative reproduction. The very characteristic feature is presence of amylum stars. The basal nodal cells develops a star-shaped aggregation of cell filled with this star shape. Amylum stars, that is why I call amylum stars. Bulbils. Now on the rhizoids of Kara, see rhizoids. So question arises, where the bulbils are from? Rhizoids. Develop spherical bulbils. Then we have amorphous bulbils, they are present at the lower nodes. So be careful, it will come as a bulbils or amorphous bulbils, right? A very important part we are starting. So be attentive. Sexual reproduction. The male sex organs are called antheridium or globule, whereas female is called nucleus. Don't confuse it. But female, we have M, and the next word to M is MN, nucleus. Both the these organs are macroscopic. I already told you this is a characteristic feature because we started with it. It is basically the multicellular filament, and they are we started with the multicellular and macroscopic, right? So now we are coming to the antheridium or globule. They're, as I told you, they are macroscopic, spherical, red or yellow structure, outer shield cells, right? They are covered outside by means of outer shield cells. A rod-shaped cylindrical cells arise from the center of each shield is known as manubrium. So we again go back to our book. I will show you. We termed amylum stars, and this is the second is the bulbils, and then is the morphal bulbils, and then we are coming to this part, that is the basically nucleus and globule, right? Because they are produced, mostly they are produced on the same branched, they are homothallic, but if they produce on different, that is stereothallic, right? Now we are coming to the first basically explaining the globule. So globule, you will see it is covered on outside by means of the shield cells, right? And from here it arises a, I told you, manubrium, right? And manubrium, from manubrium we have these secondary capitulum cells, and from there we will get this first primary and then the secondary, first primary and then the secondary, right? So you should be very much clear about this structure. So I will do as we are doing. So then we have this one, outer shield cells, and a rod shaped, I showed you just now, a rod shaped cylindrical cell arise from the center of the each shield, known as manubrium. The question is the rod shaped center from dash, this is manubrium. Now again we are coming to the nucleus. They are also macroscopic, oval, and at the node of primary laterals. Then 5 spirally twisted tube cells, they cover the nucleus, except at the tip where there we have the 5 corona cells. So we are coming to the next. See, this is nucleus and it is surrounded by the tube cell, except at the top, the 1, 2, 3, 2 back is called corona cells, right? It has egg nucleus, then stalk, and this is the pedicel, right? So the food material, which is the 5, then food material, corona cells, then 5 food material stored is the egg is in the oil drops and starch grains. Because starch is basically present everywhere in chlorophyll. The oospores of Chara do not germinate. Very important. The oospores of Chara do not germinate. They require white or red light for the germination. So this also question arises. Now the economic importance of algae. So we know agar-agar, which is used in plant tissue culture, it comes from Gelidium, Gracilaria, Chondrus. Then carrageenan obtained from the cell walls of Chondrus crispus and Gigartiana stellata. Alginate from the laminaria. Alginate, here I want to tell you that from the alginate we will get the sodium form, that is sodium alginate, and this sodium alginate is used in making artificial seeds, that is we also call synthetic seeds, and they are very much used nowadays in tissue culture, right? Then brown algae, laminarials or kelps, they are rich in iodine. Iodine is a very good source we require. Soda, potash and alginic acid. Right? Food obtained from laminaria saccharina is known as combo. This also question arises. So this I finished. It's a big, I already told you. And next I will start, because I again tell you, revise this diagram, try to make it as many times as possible. And then we will start the next. This we already did. Revise them, particularly this parasitic algae, cryptic algae, right? So revise this, and on the subsequent day I will move to the next. I have a lot of things, that is Vaucheria is there, right? And Xanthophyce is there, Rhodophyce, Metricospermum is there, and we left with Cyanophyce, but Cyanophyce we will do later on. So okay, and take care, be indoors, be safe, bye bye, see you later.